We are starting with protein targeting to endoplasmic reticulum. When protein enters ER, it is called translocation. When protein leaves ER, it is called retrotranslocation or dissociation. This process of translocation can be co-translational or it can be post-translational. If it is co-translational, it has to be membrane bound. And such ATS ribosomes, which will be membrane bound, will start synthesizing a peptide, a protein, which is having a specific signal sequence at N terminus. This specific signal sequence is 15 to 35 amino acids long, and these are hydrophobic amino acids, so we can call this sequence as N terminus hydrophobic stretch. This N terminus hydrophobic stretch is being recognized by this SRP, which is signal recognition peptide. This SRP is a ribonucleoprotein present in cytoplasm. After this N-terminus signal sequence has served its purpose, it may be either retained or it may be cleaved or removed. If it is removed, this process of removal is mediated by enzyme signal peptidase, which is present in the membrane of endoplasmic reticulum. Now we will talk about some properties of these specific proteins which decides its fate, that where it will be translocated, whether it will be translocated to lumen of ER or it will be inserted into endoplasmic reticulum membrane. Inside the membrane, whether it will be integral membrane protein or it will be peripheral membrane protein. Okay, so there are two basic criteria for such fate of these proteins. One is specific signals and second one is physical properties of such proteins. Talking about physical properties, if the protein is water soluble, it will be targeted to lumen. If it is fat soluble, it will be targeted to membrane. And if it is water soluble and having some post translational modifications, like it will be having some lipidation motifs which will be used to attach lipids. The lipid attachment may be GPI anchoring, it may be prenylation, it may be meristylation, it may be palmitylation. Such proteins will be act as the peripheral membrane proteins. The specific signal sequence will actually determine the specific location of the targeting. If there is N-terminus hydrophobic sequence of 15 to 35 amino acids we talked about earlier, it will be targeted to lumen. If there is a specific stop sequence in between or internal stop sequence, we can say, it will be acting as integral membrane protein. If there is only single stop sequence, it will be single pass protein. If there are multiple stop sequences, it will be multi-pass protein. In multi-pass protein, if there are even number sequence, it will be even pass protein with N terminus and C terminus on the same side. And if it is odd number of stop sequences, it will be odd pass integral membrane protein with N terminus and C terminus on the opposite ends or opposite sides. This was the fundamental basis of ER translocation and how properties of these proteins will decide their fate. Now we will talk about this specific signal recognition particle, its properties, its nature and its functioning in upcoming slides. Signal recognition particle is cytosolic ribonucleoprotein. It is having both protein as well as RNA. RNA is of 7SL small cytoplasmic RNA. It is non-coding RNA itself. It is coded by RNA polymerase 3 and it shows homology with ALU sequence, which we will study in transposon. There are 6 protein subunits arranged by their molecular mass like P9, P14, P19, P54, P68 and P72. The most important one is P54 because it is having both GTP binding domain as well as GTPase activity. This SRP particle or SRP ribonucleoprotein shows 3 types of interaction. The first interaction is with this specific signal sequence of newly synthesized protein. The second interaction is with the ribosome itself, and it interacts with this ribosome through the 7SL scRNA, which we talked about earlier. This 7SL scRNA mediates interaction with the ribosomal RNA, as you can see in this diagram right here. The third interaction will be between this SRP and SRP receptor. The SRP receptor is present on endoplasmic reticulum membrane. When this interaction of SRP and SRP receptor will take place, there will be docking of this ribosome to the endoplasmic reticulum membrane. As the ribosome docks, GTP hydrolysis will take place and this SRP particle moves away, and the process of uh, translocation will begin. Now, the important point to be noted here is that this signal recognition particle consists of two domains. By ALU domain, it interacts with ribosome, and by S domain, it binds with this specific signal sequence as well as this SRP receptor. And when it binds with the ribosome, as you can see here, it causes elongation, arrest of translation. This ribosome is synthesizing the protein. When this ribosome and SRP binds with each other, there will be arrest in elongation phase of translation by this ribosome. Why is this arrest important? Why the translation process needs to be stopped? It is stopped to prevent the insertion of this protein synthesized by this ribosome into the cytosol. This protein is only meant for this endoplasmic reticulum. It should not be released in the cytosol. That is why this elongation arrest is important. And when ribosome docking takes place, the GTP hydrolysis of this SRP. No, what is the site of this GTP hydrolysis? Where is this GTP bound in the first place? This GTP is bound with this P54 protein subunit of the protein part of this ribonucleoprotein, and when this GTP hydrolysis takes place, the SRP particle dissociates and moves away. When this particle moves away, the process of translation begins or resumes, and the process of translocation will also take place or will also start right away. Let's talk about SRP receptor. SRP receptor, you can see, is present in this endoplasmic reticulum membrane. This receptor is heterodimer, means consist of two subunits and both the subunits are different. One subunit is embedded in the membrane, as you can see, and other subunit is lying in the cytosolic phase. Both these subunits, the cytosolic subunit is termed as alpha, and this embedded or membrane helical subunit is termed as beta subunit. Both the subunits are having the GTPase activity and GTP binding capability. The prokaryote homologue to this SRP receptor is FTSY, which is present on plasma membrane. And for this ribosomal interaction to happen, this interaction right here, both this SRP receptor as well as SRP protein, ribonucleoprotein, both of them needs to be in GTP bound form, and this SRP receptor is bound with GTP with this alpha subunit, whereas this SRP protein is bound to GTP with this P54, which is a protein subunit out of 6. Here is the basic idea of ER Lumen translocation. Membrane free ATS ribosome comes. It starts synthesizing a peptide with N-terminus specific hydrophobic patch. This patch is recognized by the SRP protein. SRP protein recognizes this patch and interacts with SRP receptor. SRP protein, when binds with a ribosome, it induces the translation arrest, and when it interacts with the receptor, ribosome binds to this membrane and it results in ribosome docking. SRP moves away due to GTP hydrolysis. The protein will start translocating into lumen, as well as the translation process has also begun by removing this SRP particle. SRP particle will recycle into a new cycle of this ER lumen translocation. SEC61 is heterotrimeric protein complex, and prokaryotic analog is SEC YAG complex. It is having two types of gates. First is this vertical gate. The protein enters from cytosolic phase, will move through its this channel spore and enters ER lumen. This is known as vertical gate. Whereas there is one more horizontal gate. If the protein is to be inserted in the membrane of endoplasmic reticulum, the protein is inserted and this protein moves laterally through this lateral gate and will be translocated in the membrane of ER. The first one shown in green is alpha, second one shown in orange is beta, and third one shown in blue is gamma. The alpha subunit acts as translocation channel. The vertical gating system and the whole translocation channel is comprised of this alpha 1 subunit. The beta subunit is stabilizing the whole complex, and the gamma subunit is assisting the gating system and assisting ribosome interaction. While this translocation has to occur, the ribosome is the one which is pushing the protein downward so that the backsliding of protein can be prevented. One more function of this lateral gate, this is the specific signal sequence. As in the beginning I talked about, this sequence can be cleaved off by a specific signal peptidase enzyme. So this signal has to be cleaved off by that enzyme and moved laterally to membrane where it can be degraded, and final protein is lacking this segment. This is also the function of lateral gate, that whole protein will be translocated by vertical gate, but this specific signal sequence will be translocated by horizontal gate, and further it will be degraded in this membrane by enzymatic action. This is the overview of Sec61 luminal translocation. So we start with membrane-free ATS ribosome. It comes and peptide translation begins. There is N-terminus specific sequence of hydrophobic amino acid, which is 15 to 35 amino acids long. There is first recognition by SRP. SRP interacts with ribosome, leads to translation elongation arrest. When SRP interacts with the SRP receptor, ribosome docking will take place. As ribosome docks, GTP hydrolysis will take place. SRP dissociates. The translation elongation will resume. When translation elongation resumes, there will be opening of this vertical gate. This opening is triggered by sequence recognition by Sec61 complex itself, which initiates the start transfer signal. That is why in the beginning I also said that this signal is sometimes called as start transfer signal, because it is also recognized by the translocone channel, and the peptide is co-translationally entering the Sec61 channel. Now, the peptide is having normal amino acids as well as the N-terminus hydrophobic signal stretch. The normal protein has to enter the lumen, but this signal sequence is cleaved off. It is cleaved off by signal peptidase, Sec61 associated protease. It is associated with the Sec61 translocone itself, and it leads to cleavage and removal of hydrophobic signal sequence at N-terminus. When this cleavage has completed, the lateral gate of Sec61 opens and the cleaved sequence has to be transferred to membrane, where it will be subjected to enzymatic digestion. Now, let's come to some assisting protein. There is one assisting protein which is binding immunoglobulin protein. It is a kind of chaperone. It belongs to HSP17 chaperone family. It is located in the lumen of endoplasmic reticulum. As you can see, the protein is entering the ER lumen. This BIP protein will associate itself with this binding, this entering protein. This protein is prevented from misfolding. That is the major function of this BIP, is to prevent the misfolding of currently entering protein. It also assists in the correct folding of entering protein. Post-translational translocation, this BIP also helps in preventing the backsliding of newly entering, post-translationally entering protein. It is also useful in the ERAD mechanism, which directs terminally misfolded proteins to proteasomal disintegration. Protein works by ATPase cycling, which we have already seen in previous videos. Overexpression of BIP is hallmark of cancer cells, and it helps survival under stress conditions as well. Mutation impairs the BIP function, which relates to ER stress diseases, neurodegeneration. The free ATS ribosomes can either have a strong N-terminus sequence. If it is having a strong 15 to 35 amino acid long N-terminus sequence, it will be delivered co-translationally, and ATS ribosome docking will take place. SRP protein, SRP receptors, Sec61 complex, Sec63, and in most of the cases BIP will also be involved in the translocation of such co-translational translocation, as you can see in this diagram. In case of post-translational translocation, the problem is that the peptide synthesized is either very small or it is having a very weak signal sequence which is either not 15 to 35 amino acids long, or which is not that much of hydrophobic sequence, or it lacks the signal sequence altogether. In such a case, post-translational translocation will take place, and in case of post-translational translocation, SEC61 is not sufficient on its own. The other two most important proteins which will be allowing this transfer are SEC62 and SEC63, along with BIP, in which BIP ensures that there will be no backsliding of this protein. As you can see here, different proteins are involved in post-translational translocation. Here's the subtle comparison of co-translational translocation and post-translational translocation. In case of co-translational translocation, we have a ribosome pushing down this protein, preventing backsliding. We have cleaved off ER sequence which will be transferred through lateral gate on this membrane and will be degraded eventually. In case of post-translational translocation, you can see SEC61 complex is associated with SEC62 and SEC63 complex, and there is association of BIP proteins which are working through ATP cycling mechanism, and these BIP proteins are preventing the backsliding of this protein and ensuring that this protein is pulled into the lumen. Let's come to SEC63. SEC63 is 3-pass transmembrane protein in which C terminus is cytosolic, N terminus is luminal. There is J domain that interacts with these chaperone proteins like BIP, and which facilitates in post-translational import. The role of SEC63 is majorly in post-translational translocation. It stimulates the BIP ATPase activity, which ensures the pulling of protein into ER lumen. It acts as a molecular ratchet, which is pulling the chain in and preventing the backsliding. SEC63 mutations lead to polycystic liver disease in some cases as well. In case of SEC62 or even pass protein, both N and C terminus are cytosolic, and its role is in post-translational substrate recruitment to SEC61. It works with SEC61 and SEC63. It acts as initial receptor for signal sequence of post-translational protein, and mostly this signal sequence is on C terminus domain. It helps docking of substrate polypeptides to SEC61 channel, and it works with SEC63 as a complete functional unit. Mutations in SEC62 is associated with the cancer progression, specifically in lung cancer, prostate cancer, and thyroid cancer due to stress pathway deregulation. In case of post-translational translocation, the energy is provided by ATP hydrolysis by the BIP protein, which ensures the unidirectional transport and prevents backsliding. The energy source for co-translational translocation is not that much of clear, but it is very much similar to this post-translational translocation, just like we said, the ATP hydrolysis with respect to initiation of that process. Here is the chart showing all the type 1, type 2, type 3, type 4. Another two types are tail anchored protein, which are mostly transferred post-translationally, and GPI anchored proteins as well, which are manually transferred after they are being transported inside this membrane. We will see this mechanism. The specific examples are also given right here. You can study on your own. Here is the structural composition of these endoplasmic reticulum transmembrane proteins, like type 1 having specific cleavable signal sequence and a stop sequence. Type 2 is having N terminus cytosolic, a specific stop sequence. Type 3 is having N terminus luminal, C terminus cytosolic, and a stop sequence. In case of type 4, similar kind of arrangement can be seen for type 4A and type 4B as well. Here is the comprehensive table for the comparison of all the types of ER membrane protein classification. Here we talked about the N-terminus location, whether it is non-cytosolic or cytosolic. We talked about specific signal sequence, whether it is present, it is absent, it is cleavable, it is non-cleavable. We talked about stop transfer signal sequence. You can see key determinants like positive charges on different sides, which determine For example, here you can see this is the interesting one, that the positive side determines the cytosolic location. The positive side always prefers cytosol. It is also known as inside rule, inside positive, inside rule. Here, the main difference between type 2 and type 3. In case of type 2, positive side is towards N terminus, so N terminus will be cytosolic. In case of type 3, positive charge is towards C terminus, so C terminus will be cytosolic. So positive charge also determines the localization of N terminus and C terminus as well. In other features, we shown some important examples and other attributes related to these types of proteins. So in short, type 1 and type 3 proteins have N terminus in lumen. Type 2 and tail anchored proteins have N terminus in cytosol. Type 4A and type 4B differ in N terminal orientation. Now we will talk about how these different types of transmembrane proteins are inserted into endoplasmic reticulum membranes. Let's start with type 1 proteins insertion. So here is the basic structural arrangement of type 1 proteins. It is having cleavable signal sequence. So let's look at the diagram itself. The ribosome is pushing the protein downward. The signal sequence is being recognized by Sec61, allowing the protein to transfer into lumen. There is a signal peptidase which is associated with Sec61. This signal peptidase will chop down this signal sequence. The lateral gate opens and this signal sequence will be transferred to membrane itself and will be degraded proteasomically. The rest of the protein will be transferred to lumen until we reach the stop sequence. The same stop sequence be shown here. When the stop sequence came, there are hydrophobic amino acids. Now these hydrophobic amino acids cannot enter the lumen because lumen is hydrophilic. So these hydrophobic amino acids will stay in the membrane. Lateral gate opens again and this protein will be translocated into the membrane itself and will be inserted to membrane with C terminus in cytosol and N terminus in lumen. Let's talk about type 2 transmembrane protein insertion in endoplasmic reticulum. Here is the structural map of type 2. The positive charge is toward the N terminus, means N terminus has to be cytosolic. This is known as positive inside rule. As you can see, the protein is synthesized by the ribosome. The positive charge is towards the N terminus, and the protein starts entering the lumen. The hydrophobic sequence or stop signal is reached. This stop signal consists of hydrophobic amino acids. The protein no longer can go into lumen itself. The lateral gate opens. Stretch of sequence is being translocated inside the membrane and the protein is adjusted accordingly, where N terminus is towards the cytosol and C terminus is towards the lumen. In case of type 3 transmembrane transport, you can see the positive charge is towards the C terminus. So C terminus has to be cytosolic. The similar phenomena here, the translation is occurring, which is synthesized. The hydrophobic amino acid will stuck in the membrane. They will be translocated through the lateral gate. Protein will be integrated in such a way that C terminus is cytosolic and N terminus is luminal. Now let's come to type 4 transmembrane protein insertion. Again, the positive inside rule will apply. The positive charge is toward the N terminus, so final protein N terminus will be on the cytosolic side. But what will happen that there are multiple stop signal sequences, one right here, second is here, third is here. When first stop signal is reached, it cannot move to lumen. It will be translocated through lateral gate. Right, you can see here. Then reaches the second stop signal sequence. It will be translocated through the lateral gate as well. Now you can see two helices in the membrane. Then it reaches the third sequence, which will again be translocated by lateral gate, and we have 3-pass transmembrane protein synthesized in the similar way. This was the type 4 protein translocation into the ER membrane. Here is the comparison of type 4A and type 4B proteins. Like N terminus location is cytosolic in type 4A, it is luminal in type 4B. Their first hydrophobic sequence function, like in type 4A, it functions as anchor sequence, whereas in type 4B it functions as signal anchor sequence that inserts N terminus into lumen. Whereas in case of type 4A, it inserts this N terminus into cytosol. the positive rule is followed, no such rule is followed in case of type 4b. We have certain examples like GLUT1 and GPCRs which follow type 4a localization mechanism, and we have CFTRs and some other GPCRs which follow the type 4b translocation mechanism. Now let's come to GPI anchored proteins. So ATS ribosome synthesizes these proteins. The enzyme involved is GPI transamidase, which recognizes the specific omega site near the C terminus. So the main sequence here is C terminus hydrophobic stretch, and it acts as signal for GPI anchor attachment. Now GPI transamidase recognizes a specific omega site near the C terminus. The GPI anchor consists of this phosphatidyl inositol, which is lipid tail inserted into membrane. We have a glycan core. We have ethanolamine phosphate, which attacks and makes amide linkage as well. Now there is cleavage of C-terminus hydrophobic signal sequence, covalent linkage of C-terminus of protein to a pre-assembled GPI anchor, and the formation of amide bond will take place between ethanolamine phosphate and C-terminus omega site residue of the protein, and further there will be maturation and transport like glycosylation, and further transport will occur. So this is the membrane inserted protein. C-terminus is cytosolic, and terminus is luminal. Here is the inositol phosphate which attacks the amino acid. As it attacks the amino acid, the transamidase enzyme catalyzes this reaction and it forms a amide linkage, amide bond here, and the protein will be linked to these fatty acyl chains. This diagram exactly shows the same thing, that this is the ethanolamine and the phosphate attacks this sequence of proteins. This is attached itself to an oligosaccharide, and finally this protein will be attached to a GPI anchor linkage. Now we will talk about ER membrane insertion in case of post-translational proteins. ATS ribosomes, which are membrane-free ribosomes, they can perform SRP-mediated docking and go into co-translational insertion, or they lack a strong signal, they lack short peptide, and they will go post-translational insertion. In case of post-translational insertion, we have tail-anchored proteins, we have multipass proteins. Multipass proteins can be inserted by Sec61, 62, 63 and BIP mechanism we talked about earlier, and if the Sec61 cannot handle this insertion, the ER membrane complex insertase enzyme complex will help in this insertion as well. In case of tail anchored proteins, we have a pair of different chaperones like HSP70, HSP40, S-gata, which binds to the C-terminus specific domain. We have TRC40, which is GET3 ATPase in yeast. It binds to the C-terminus domain. We have TRC40 substrate complex, which is GET1, GET2 complex in ER membrane. This GET3 will interact with this GET1, GET2 complex. There will be ATP hydrolysis and release of substrate in ER membrane. That's how the protein translocation will take place. Here are the more details on these chaperonic proteins like HSP7040, they function, what is S-gata. You can go on your own for this. We also have membrane receptors like GET1 and GET2. We are directly moving on for the diagram of this translocation. There is translation of protein. This protein is having C-terminus specific sequences, which is specific TMD sequence. This protein is bound by these chaperones to prevent misfolding. Also, it is interacted by this TRC40 or GET3 complexes, and these complexes will bind to this C-terminus domain, and these complexes will bring this protein to GET1, GET2 complex. In mammals, this GET1, GET2 is known as WRB or CAML complex. The docking of this protein will take place and it will be transferred to the membrane itself. We will talk about what is the fate of misfolded endoplasmic reticulum proteins. The recognition of misfolded proteins is important. When the proteins or misfolded aberrant proteins are recognized, they are retargeted to ERAD machinery. In case of ERAD machinery, they will be degraded by proteasome through ubiquitinylation process, and retrotranslocation will take place, and their final degradation, proteasomal 26S proteasomal degradation will take place. Now we will see diagrammatically how this process works. As you can see, this is some kind of misfolded protein. the chaperone attaches with this misfolded protein to contain it. There are disulfide isomerase. This disulfide isomerase break disulfide bonds to fully unfold this protein. Chaperones are the ones which are keeping this protein in unfolded state. There are lectins, as you can see green one here. Lectin recognizes the N-link glycosylated proteins which mark that these proteins are improperly folded. Now these three interactions will take place. One is chaperone which prevents misfolding, or there is disulfide isomerase which is breaking disulfide bonds to further unfold this protein, and there is lectin which is recognizing the N-linked glycosylation. Now this lectin has high affinity for this protein translocator complex. This protein translocator complex is tightly associated with E3 ubiquitin ligase. When the protein is translocated, this E3 ubiquitin ligase will ubiquitinylate this unfolded or misfolded protein. Here is the role of triple adenine ATPase. Kinase will remove this glycation on this protein. The protein will be polyubiquitinated, and this polyubiquitinated protein will be moved to 26S proteasome degradation system and will be degraded there. Now we will talk about a very interesting system which is unfolded protein response system. In endoplasmic reticulum, folding of secretory and membrane proteins takes place. If this protein folding is abnormal or endoplasmic reticulum is under stress, the endoplasmic reticulum stress includes hypoxia, glucose deprivation, certain mutations, effect of toxins, there must be some kind of viral infection or high secretory load. In such a case, to relieve from such stress or to solve the problem of unfolded proteins or misfolded proteins, the UPR system will be activated, and UPR system activation will ensure the restoration of balance. To understand UPR system, we must understand that on the membrane of endoplasmic reticulum, there are certain specific receptors which sense the normally folded proteins as well as misfolded proteins. And when they sense that proteins are misfolded, they will take action. They will start downstream signaling and will lead to effector response. You can see here is a generalized structure of sensor present in endoplasmic reticulum membrane. On the upper side we have cytoplasm, on lower side we have endoplasmic reticulum lumen. Now this sensor protein is having three domains. First domain is cytosolic domain. The cytosolic domain is having kinase activity. Mostly it is serine threonine kinase. It is having RNase activity. It is having activity to act as some kind of transcription factor and perform their function as well. We have transmembrane helical domain which is present in alpha helix form in this plasma membrane, and we have luminal domain. In case of luminal domain, we have stress sensing domain, we have specific domain to recognize the misfolded proteins as well. Now normally what happens when everything is normal, ER is not under any stress, the BIP proteins which are chaperones, they have high affinity for this sensor and they bind to place in a luminal domain. They have specific binding site in the luminal domain. When everything is normal, proteins are normally folded, BIP is having high affinity for these stress sensors and it binds with these sensors, and whenever BIP is bound with this stress sensor will be inactive. Now let's see the contrast. When protein is misfolded or some kind of stress is going on, ER stress is going on, there might be hypoxia, there might be glucose deprivation, there might be any kind of viral attack, there are misfolded proteins. When the misfolded protein concentrations start rising, the BIP will show higher affinity towards these misfolded proteins and will bind with these misfolded proteins, which will make the stress receptors free. When these stress receptors are free from these BIP proteins, they become active. And when these stress receptors will become active, they start dimerization process. And when these stress receptors dimerize, this dimerization brings together two cytosolic domains, and when these two cytosolic domains are brought together, the cytosolic domains, as we described earlier, the cytosolic domains have kinase activity. So these uh, domains perform auto cross phosphorylation, which means that this domain phosphorylates this and this domain phosphorylates this. This auto cross phosphorylation will lead to effector response, and there will be a variety of effector responses we will talk about in upcoming slides. Now, second mechanism is like which is independent of this BIP binding. Suppose this BIP is already uh, being dissociated and bounded to these misfolded proteins. Now, these receptors also have specific site for binding of the misfolded protein. The misfolded protein segments will bind to these specific sites, which are most common in IRE1 stress receptor. We call that protein binding domain. It directly recognizes the misfolded protein. When it recognizes the misfolded protein, again it will initiate dimerization. When it will initiate dimerization, there will be again cross phosphorylation, which means this subunit phosphorylates this and this subunit phosphorylates this. When this autophosphorylation takes place, there is conformation change, there is increase in kinase activity, there is increase in RNase domain activity, and there will be a specific effector response. Now let's see what kind of effector responses will be there. As you can see, there are three stress receptors: IRE1, PERK and ATF6. All these stress receptors have been activated and they will finally produce some kind of transcription factors, and overlapping targets of these transcription factors will produce gene products that improve the protein processing capability. All of these collectively leads to activation of genes to increase protein folding capacity or endoplasmic reticulum and protein degradation apparatus. It improves the functioning of proteasomal degradation. It improves the quality control of protein synthesis. It improves the quality control of protein folding as well. That is the main effector response of all these proteins. I can make separate videos if you want on all these mechanisms separately. Here is the cytosol, here is ER lumen. In IR stress receptor, the BIP protein has already been removed due to its higher affinity with misfolded proteins. The misfolded proteins directly bind to this protein binding domain of IRE1 receptor, which will lead to dimerization of IRE1 receptor. On dimerization, it will cross phosphorylate, auto cross phosphorylate, and this auto cross phosphorylation will regulate the specific transcription regulator, which will go to nucleus and initiate the formation of specific proteins. Inside nucleus, it codes for chaperone mRNA, which codes for chaperone proteins. These chaperone proteins will enter the endoplasmic reticulum lumen, and this chaperone protein will assist the folding of these misfolded proteins. They will bind to these misfolded proteins and allow the proper folding of these misfolded proteins. When these misfolded proteins are properly folded, there will be lesser of misfolded protein, so BIP will be bounded to these stress receptors and these stress receptors will be deactivated. Here is the comparative table of these stress receptors. You can see the final outcomes like protein folding capacity will be increased, ER expansion will be there, misfolded proteins will be cleared and degraded in proteasomal system. These are specific for different stress receptors as well, and they will ultimately ensure the survival and a reduction of stress and return of normalcy.